I'm very happy to welcome you all to this workshop that we're very happy to be able to have put together over the last weeks and month. Today we're going to talk about bioinformatics in cancer research, and I hope that we'll try to elaborate on the status of technologies that we have available in Tübingen and with the help of very well-known guest speakers, we'll also look into application areas, so areas of cancer research where data-driven approaches play a big role. Now, why do we think this is all important? Over the last years, there has been a big hype around big data, and we've seen big data is there to solve the cancer problem. At least it's stated by mostly public media. And as you can see here, the public media really jumped on it. And a couple of years back, the Time was talking about one of the big players, namely Google. If those players, industrial players, cannot jump in and to solve major biomedical problems. I don't think this is all that true, and I think academia is very well set to dive into that game. And effectively, why this is possible, because we have now means to digitize samples, cancer being one example that we discussed today, that of course also holds true for other diseases. We have now means to really do high throughput sequencing, do high throughput mass spectrometry. Literally, we are able to profile all the omes from the genome to the metabolome. This was mainly triggered by the introduction of newer generation technologies since about 15 years with respect to sequencing and mass spectrometry. And effectively, it's not only that we are able to generate data ourselves, and we have very good facilities here in campus and very good research groups that do generate these data. It's also the public domain has a lot of data available. And as you can see here in that graph, the number, and this is just a simple query in PubMed. And as you can see, the number of multi-omic studies is really increasing. And all these data sets are available that we can use for our research with the bioinformatics methods or the data science methods in place. That's why I think it's not only that we can generate the data, but also the means to use publicly available data for research projects at hand. Now, as I said, the data is there and we can generate it, yet the problem is not solved yet, because it's clear that the data that we generate today, we need to make sure that we also can reuse it sustainably. And there has been an interesting report back in 2014 that literally stated that 80% of the research data that is older than 20 years is not available anymore because it has not been properly annotated. We simply cannot find it anymore. So this is one thing that contains data management as an important aspect in the research that we need to put in place. On the other hand, it's research reproducibility. There has been a survey that states that 90% of the researchers, especially when it comes to computational analysis, fear a significant reproducibility issue. So whatever we do on the computational side, we also need to make sure that it's very transparent and that we can ensure that our methods are also reproducible. Now, over the last years, there has been the introduction of the notion of FAIR, and we've also had a contribution that elaborates on FAIR data and also FAIR software. So literally to overcome the problems that we have in reproducibility when it comes to computation and also when it comes to data management, the FAIR paradigm, that's literally now what all the funders want to see in our proposals if we submit it, that we make sure that our data and software is fair, which means that we need to make sure that we can find the data. So we need to annotate our data sets properly, that it's really findable, and then also provide the applications that allow us to find the data. And as you may imagine, the larger the data sets grow and the more we have of them, the more difficult this gets. Now, data also needs to be accessible. It, of course, doesn't make too much sense if we keep to have the silos and that we close the silos and only use them for a very small set of researchers that have access to the data. So we need to come up with technical means that make data accessible, yet that, of course, does not mean that it's accessible directly to the world, especially when we deal with patient data. There, of course, needs to be mechanisms that make sure that authentication and authorization are in place. Technically, it has been shown that the interoperability is a major issue. So it's not necessarily given that if we run computation analysis in one lab, that we can also run them in another lab, because we may not be able to read in each other formats. We may not be able to work on each other's infrastructures and so on. Then the last letter of FAIR is the R, which denotes reusability. And that, of course, is very important. The data that is available in the public domain, that needs to be reusable in order to make sure that we can really do big data-driven research, that we do not need to generate all the data again for each and every research project, but make use of the existing data. If we put these four letters together, then we usually like to add an additional R and say that we'd also introduce the notion of reproducibility that we consider very important for computation analysis in cancer research. And using the four letters, I think the reproducibility part is easier to implement as if you don't have FAIR yet. Now let's come to the workshop, since I've only a couple of minutes for the introduction. So what we're going to do today. The workshop comprises lectures and relevant applications from the field of cancer research. And we have several distinguished guest speakers that from the Tübingen campus run major areas in cancer research. And we'll just look into the agenda in just a minute, just to give you an additional summary of what we're going to do. We'll try to present you bioinformatics methods, but not only methods, but also research applications that where the methods can be used. Then we'll look, and this is simply possible, as I mentioned, because we have seen major breakthroughs in data generation, but also in the analysis. We'll look also into data management, namely FAIR, and how that could be applied to cancer research. Literally, it's not only omics, we have also significant component on imaging, which will be in the afternoon. So to introduce the speakers, I'm almost done with my 10 minutes. Then we'll have Juliane Walz, which she's a group leader in the immunology department here. And talks about peptide-based immunotherapy. So is Markus Löffler, who also has an immunotherapy talk to present. Then we'll have a cubic contribution. Sven will talk about FAIR data management, followed by Friederike Hansen. She will talk about portability and reproducibility, especially of computational pipelines as applied to in cancer research, most likely also a bit of cloud computing here. Then we'll have from the genetics department, Nicola, who runs the facility down there, and he will talk about the technologies that we have available in Tübingen to generate large scale and high throughput sequencing data. Then we'll have a coffee break and followed by a talk by André Martins. He's leading a group in the imaging department, and he also obviously talks then about imaging tissue physiology. Then again, we have a cubic contribution by Louis Kuhn. So Louis Kuhn built up lately infrastructure and also analysis methods for imaging data, and probably he'll also go a little bit into machine learning there. At the end, we'll try to conclude the workshop and hopefully also have some discussion in place. As you know, probably from good old in-person workshops, I mean, it's usually very good if we use these kinds of events to also interact. So despite the fact that we needed to opt for the virtual setting, please feel encouraged to still interact. We have the coffee breaks for that. We also have some time at the end of the workshop, and we also have separate rooms available for doing that. As I said, after the workshop, we'll also be there and hopefully use the time for interaction. So before I hand over to Juliane, I'd like to take the opportunity to thank all speakers that present today. It's really great to have you here, and we really appreciate the time you take to come to that virtual event. Hopefully at some point we can do it in person again. I'd also like to thank the organization committee, namely John, Sabrina, Luis, and Daniel, and Marissa, Kevin, and Laurence, who will take over the moderation for the individual sessions. So those will be the chairwoman and chairman. Okay, that's it from my side. I'm really curious for what's going to happen over the next hours, and I'm really looking forward to the workshops. Thanks all for attending, and we're looking forward to the talks.